Hello everyone. So I just want to do a quick mic test. I'm actually sitting in my car in the parking lot of my son's school. He has an event this morning and I have 30 minutes. So I thought, hey, let's record this part of this video real quick. I'm going to do a quick sound check to make sure that the car is not being overly loud on the video. Okay, all seems good. So we're going to go ahead and jump in. Now, while there are lots of different reactions that take place on the cellular level, when it comes to being able to harness and harvest energy, the two we will focus on are cellular respiration and photosynthesis. So this video is going to be over cellular respiration. So the organelle that cellular respiration is going to take place in, or at least most of the steps of cellular respiration in eukaryotic cells, will take place in the mitochondria. Mitochondria is plural, mitochondrion is singular, just for reference. So remember, this is a membrane-bound organelle that we find in eukaryotes. Prokaryotes don't have it, but they are still capable of doing some kind of reaction that allows them to harness energy. So that is going to really happen along their plasma membrane. That is going to be their site of metabolism. So when we talk about cellular respiration, it comes in two forms. We can reference aerobic respiration and anaerobic respiration. The big deal with aerobic respiration, so I want you to think of aerobic exercise as an example. This is going to be the type of respiration that happens in all eukaryotes and can happen in prokaryotes. And the big deal is it happens in the presence of oxygen. Oxygen is a reactant in the reaction that allows us to make ATP. So without oxygen, aerobic respiration cannot take place. Anaerobic respiration is the opposite. It's going to be the cellular respiration that happens in prokaryotes, does not happen in, well, it does happen in eukaryotes, but in a very specific way, which we'll talk about. And a molecule separate to oxygen is used in the ATP making process. So it happens without the presence of oxygen. So redox reactions are coupled reactions, meaning they go together, and they're opposites. They're made up of two separate steps or two separate reactions, if you will. The first one is called oxidation, and in oxidation, electrons are removed from a substance. And when that happens, we say that the molecule or the substance has been oxidized. The opposite but coupled reaction to oxidation is reduction. And in reduction, it is the addition of electrons to a substance. When the substance gets those electrons, we would say that that substance has been reduced. And this one in particular sounds weird because it's the opposite of what the word suggests. But in reduction reactions, we are adding electrons, and in oxidation reactions, we are taking electrons away. Okay, so again, in oxidation, it's the removal of electrons. That substance becomes oxidized. In reduction, it is the addition of electrons. That substance becomes reduced. So here is a graphic to help with redox reactions, and it's important to remember that these are going to happen at the same time. So in your reactant, something is going to lose electrons, and excuse me, and it's going to get oxidized. Something is going to gain electrons, and it's going to become reduced. Okay, so here we're looking at combining methane with oxygen, and the result of that is making carbon dioxide and water. So to make the carbon dioxide, methane is going to become oxidized. It is going to lose those hydrogens, okay, and it becomes CO2. Then the oxygen is going to become reduced. It is going to gain some hydrogens from that methane and get extra material added to it to make it reduced. So the oxygen becomes water. So it's really important that you know that our ultimate source of energy for 99% of all living things is going to be the sun. There are a couple of things that use chemicals because of where they live. They can't access sunlight, but the majority of the world runs on solar power. So the main reaction involved with capturing energy from the sun is photosynthesis, and it's where we capture solar energy and it gets converted into chemical energy. That's really a form of a organic molecule. Normally it's in the form of glucose. So photosynthesis is the process by which we make glucose using energy from the sun. The opposite reaction to photosynthesis is cellular respiration. And in cellular respiration, we're going to take that glucose molecule that we got access to from photosynthesis, and we are going to oxidize it. And when we do that, we're going to release energy that we can then use to power our bodies. Now, all of these two major reactions, photosynthesis and cellular respiration, are multi-step processes, but they're also examples of redox reactions. Okay? So in cellular respiration, the cell is going to make ATP, and it's going to do so by a process known as oxidative phosphorylation. And we'll talk way more about that when we get to that point. The energy that we release when we oxidize these glucose molecules is captured and temporarily stored as ATP until your body needs to use it to power whatever action it is currently doing. So both photosynthesis and cellular respiration are based on energy transfers. And this utilizes electrons, the same electrons that we talked about when we talked about the structure of an atom. So electrons have the ability to gain energy, and they also have the ability to lose energy. And they lose energy as they move from molecule to molecule, what we call donor molecules to acceptor molecules. Normally, the electrons that we're harnessing for these reactions are going to come in the form of a hydrogen molecule. Because remember, hydrogen just has the one electron, so it's easy to use that electron for various things in the body. The energy that these electrons release as they move around is going to be free energy, which means that we can harness it to do work. So in cellular respiration, we are making ATP by moving a bunch of electrons around, harnessing their energy to do the work of making or synthesizing ATP. So this diagram shows the relationship between photosynthesis and cellular respiration. And notice it's a cycle because, when you thought you had your laser pointer. So notice it's a cycle because they're constantly circling around each other. And we have a saying in biology that, the products of photosynthesis become the reactants of cellular respiration. So in photosynthesis, I'm starting off with carbon dioxide and water. I'm going to get energy from the sun. I'm going to do the process of photosynthesis. In doing that, I'm going to make some oxygen, and then I make glucose, which was my ultimate goal of the reaction, and that's photosynthesis. I go from electrons at very low energy levels to making a molecule that has electrons at very high energy levels. For cellular respiration, I'm starting off with glucose, which I'm going to break down into its component pieces. As I do that, I am making ATP, which is the goal of cellular respiration. I'm also going to utilize some oxygen in order to make my largest amount of ATP. And as I do the process of cellular respiration, I'm going to make water and make carbon dioxide. So I'm going from high energy electrons to low energy electrons. So this equation hones in on what parts of cellular respiration the redox situation is taking place. So I start with glucose and oxygen as my starting material. I am going to lose electrons from glucose. I'm going to lose atoms from glucose and take it down to carbon dioxide. So that's my oxidation, the loss. And then I'm going to take my oxygen and add hydrogens to it, so the gain, and that is reduction. Okay? For cellular respiration, my reactants are glucose and oxygen. My products are carbon dioxide, energy, and then, of course, the ATP. Carbon dioxide, water, sorry, and then the ATP, which is the form of energy. So this diagram shows cellular respiration in relationship to the organelle that's associated with cellular respiration. And specifically, this is cellular respiration in eukaryotic cells. Okay? So wherever there is a number, that is a step in the cellular respiration process. So even though we always talk about cellular respiration occurring or happening in the mitochondrion, recognize that the very first step, step 1, is outside of the mitochondrion. So really quick, we are going to talk about what they are, like name them, and why they're located where they're located, what's needed for them to occur. So, step 1 is the process of glycolysis. It happens in the cytosol. Or sometimes you'll see them say in the cytoplasm. The difference between the cytosol and the cytoplasm is just the liquid in the cell. The cytoplasm is the liquid and everything that floats in it. Okay, so all the organelles, proteins, all of that. Glycolysis happens without oxygen. Okay, and that's why it happens in the cytosol, because in our earliest living things, there wasn't oxygen in the atmosphere. They still needed a way to harness energy, so they used the process of glycolysis for that. Step 2 is known as the link reaction. Kind of happens in between the first 2 layers of the mitochondrion. Step 3 is known as the Krebs cycle, and it happens in the liquid known as the matrix of the mitochondrion. And then step 4 is the electron transport chain, and it actually happens on the inner membrane. All right, so that is cellular respiration in a nutshell. So before we actually talk through cellular respiration, we need to address how exactly it is that ATP is made or generated, okay? So the process involves the movement of a phosphate group. If you think back to what ATP looks like, it looks just like a nucleic acid nucleotide, but it has these additional phosphates. Okay, kind of like that. So here is my base, adenine, cytosine, guanine, thymine. Here is my sugar, and then normally for a nucleotide, there's 1 phosphate, but ATP is what we call a modified nucleotide and has 2 additional phosphates. So how do those phosphates get where they're going? Okay, because for me to have ATP, I have to have 3 phosphates. Well, that process of adding a phosphate is known as phosphorylation. And there are 2 versions of phosphorylation. There is substrate level phosphorylation, where I'm directly making ATP using an enzyme. And the job of that enzyme is to take ADP and transfer a phosphate to it from a substrate. Okay, so I'm taking a phosphate off of something and adding it to ADP. And it's done by an enzyme. That is substrate level phosphorylation. The other way to make ATP or to generate ATP is known as oxidative phosphorylation, and that is more indirect, okay? So it's the making of ATP because a bunch of ions are moving, giving me energy to add my phosphate to my ADP, okay? For oxidative, the key part of that being that oxy section of the word, for oxidative phosphorylation to occur, I have to have oxygen. And we'll talk about why. Here it says that it's the final electron acceptor, which is true, but we'll talk about what that means later on. Okay, so for oxidative phosphorylation, I must have oxygen. But for substrate level phosphorylation, I don't need to have oxygen. So think back to that picture we just labeled together where glycolysis happened outside of the mitochondria. And I told you no oxygen needed. Glycolysis is doing substrate level ATP production. The rest of the mitochondrion is going to do some substrate level and some oxidative. Okay, so here's another schematic that shows the carbon cycle. No, sorry, thinking of the high schoolers. Here's another schematic that shows cellular respiration and the 4 stages. So glycolysis is stage 1. Don't know why this is kind of out of order a little bit. The link reaction can sometimes be referred to as pyruvate oxidation or the formation of acetyl coenzyme A. All of these names come from molecules within the reaction themselves. And then the third one is known as the citric acid cycle. It's very commonly referred to as the Krebs cycle. And then lastly, the electron transport chain, or the ETS, some people call it the electron transfer system, so the ETS. A lot of us refer to it as the electron transport chain, ETC, but this is where the oxidative phosphorylation is going to take place. Every other time we make ATP, we're doing it via substrate level phosphorylation. So cellular respiration depends heavily on the movement of electrons. And for electrons to move, we have to have some kind of shuttle system or some kind of carrier system for that to take place. So now we're going to talk about electron carriers. We're talking about these individual pieces before talking about the actual reactions because these individual pieces play a very important role in the actual reactions. So electron carriers have a specific job. Their job is to capture or gather electrons and transfer their energy along a specific pathway that's made up of coenzymes. So we've already talked about what coenzymes are. They are proteins that have enzyme properties or abilities. So the 2 most common electron carriers that you will see in cellular respiration are NAD plus and FAD. Okay? So these guys have the ability to carry hydrogens. And because hydrogen has a single electron, they are also carrying hydrogen's electron. So when they have hydrogens attached, they are carrying the electron in the form of a hydrogen. Okay? Because hydrogen just has the one single electron. Electrons can't just bing bong and roam and, dance around the cell or the mitochondria any which way. They can't do that. They're not allowed to do that, let's say. Okay? They are usually attached to electron carriers. So if the electron carrier doesn't have the hydrogen attached, usually it has a plus sign, and that tells us that the hydrogen is missing. So like here, that FAD should really have a plus, really a 2+, because it can hold 2 of them. So here is a very simplistic schematic, if you will, of the process of glycolysis. So in glycolysis, we're starting off with the molecule glucose. And glucose is going to go from a 6-carbon molecule to making 2 3-carbon molecules. So think of, I've taken glucose and I'm going to take that molecule and split it in half. The two carbon molecules the two 3-carbon molecules that's made as a result of glucose being split are called pyruvate. Okay, so this is a really important step. Do you see this note that I have for you? Because I make 2 pyruvates, everything from here on out is in sets of 2. Okay, 2 sets of reactions, one for each pyruvate molecule that we make. So to go from glucose to pyruvate, it's a series of 10 different reactions, all of which are controlled by specific enzymes. Okay? Remember, glycolysis is happening in the cytosol. They can also say cytoplasm. And if you think of what the word means, lysis means to split or to break. Glyco means sugar, so it literally means sugar splitting. Now, while this is taking place, I still need electrons in the form of hydrogens to be moved around. So I am going to do that using one of my electron carriers. Specifically, I will be using NAD+. And when that NAD+ has an electron attached to it, it is known as NADH, and it's an electron carrier. So at the end of the reaction, at the end of glycolysis, this is what I've done. I've made 2 molecules of pyruvate. I will also make 2 of those electron carriers in the form of NADH, and they will carry my electrons to what other part of the reaction needs them. And I've made 2 molecules of actual usable ATP. So for glycolysis, there's a little bit of a weird twist. Now, at the end of the day, glycolysis does not actually produce a lot of energy. It produces a very minimal amount of ATP, only 2 molecules at the end of the entire process. But it is the process that starts off cellular respiration. The funny thing is, we need ATP to make ATP. So in the first part of the rearranging of glucose, which is the first half, if you will, of the glycolysis reaction, that glucose molecule gets broken apart and elements and atoms get shifted around, and that requires energy. So if you look at the slide, you notice up here, we are going from, hold on, sorry. I need to change the width of my pen. There we go. We're going from 2 molecules of ATP to 2 molecules of ADP, which means we used ATP. We didn't make it, we broke it down. We released the energy from it. But if we didn't do that, we would never be able to take glucose and rearrange it into 2 molecules that are 3 carbons big each. Okay? And then once this step happens, the rest of glycolysis can happen, where we actually will make ATP. Now, at the end of glycolysis, or not at the end, let's say, during this second part of reactions, we'll actually make a total of 4 molecules of ATP. So if you look right here where I'm highlighting, you will see that. But remember, we already invested 2, so we have to pay those 2 back. So when you consider it, we really only gain 2 additional molecules of ATP. So think of it as investing money. I invested an initial $500. At the end of the year, I have $1,000. And I don't really have $1,000 because I'd already given you $500. So I only made $500. Same situation here. I invested these 2 ATP so that the reaction could take place in the way that it needs to. And at the end of it, even though we make the 4 ATP here, we still have to pay back the 2 we originally made. So if this isn't banking us in terms of ATP, what else are we making that's a benefit? Well, those electron carriers that we talked about earlier, the NADH molecules, because so there are two of them. There's NADH and FADH2, but in glycolysis we only make NADH, and we do make two of them. Plus we make pyruvate, which will continue in the next steps of the cellular respiration reactions. But that NADH is going to be sent to the electron transport chain, and we will do stuff with it later. So we've already discussed the difference between the two methods by which ATP can be made, substrate level phosphorylation and oxidative phosphorylation. So we just got done making ATP in glycolysis. That ATP was made using the process of substrate level phosphorylation. So it was directly made, not indirectly, but directly made. Okay, so remember, in substrate level phosphorylation, an enzyme is going to catalyze the reaction of transferring a phosphate group from a substrate to ADP. And that's going to turn that ADP into ATP. So if you look at the picture here, so here is my ADP molecule. I know it's ADP because I have two phosphates and it's ready to go. And here is the molecule that will eventually become pyruvate. This is my substrate, okay? And the cell is getting that directly from glucose. So that enzyme is going to come in and it's going to create the situation where this phosphate can be removed from phosphoenolpyruvate, what we call PEP. And once that phosphate's removed, it gets added to ADP, making it ATP. So 1, 2, 3, I have 3 phosphates. And then this changes the identity of PEP. PEP without that phosphate is now just pyruvate, which is the end product of glycolysis, and it's what's going to start off the next set of reactions. So up next is what we call pyruvate oxidation. Some people refer to this as the link reaction, and it's because it links glycolysis to actual cellular respiration. Okay, so we know that we ended glycolysis with pyruvate. Pyruvate is now going to get oxidized, okay? And it is going to become a new molecule. So remember, pyruvate has 3 carbons. At the end of its oxidation, it will be a 2-carbon molecule that we call acetyl. Well, it has a couple of names, but the 2-carbon molecule itself is an acetyl compound. And then we're going to add some additional features to it. We're going to add a coenzyme A. And so the whole thing, the final product of this pyruvate oxidation is acetyl-CoA. Sometimes we call it acetyl coenzyme A or acetyl-CoA, same thing. This is happening in the mitochondria, so it's the very first step that's taking place in the mitochondria. We're going to make some electron carrier in the form of NADH, and carbon dioxide is produced. So in cellular respiration, every time we lose a carbon, that carbon gets coupled with oxygen and becomes carbon dioxide. This is one of the reasons why carbon dioxide is a product of cellular respiration. So you're going to have to keep up with the math here a little bit, okay? Remember, glycolysis gives us 2 pyruvate molecules once it's completely done. So each pyruvate molecule is treated in the same way. Each pyruvate molecule gets oxidized into acetyl, and then we add a coenzyme A, so it becomes acetyl coenzyme A. So we're going to make 2 acetyl coenzyme A's actually, okay? So everything is double. That's what I need you to remember. Oops. Okay, so each pyruvate is going to make an acetyl group, or acetyl coenzyme A, one NADH, and one carbon dioxide. Okay, so when you do the math at the end of pyruvate oxidation, for every molecule of glucose, we'll actually, at this point, we'll have two acetyl groups, two NADHs, and two carbon dioxides. Okay. Now, why is this reaction important? This reaction is important because we make a little bit of NADH. Those electron carriers are vital for how cellular respiration is really going to produce ATP. You're not going to see it until we get to the electron transfer chain, but that's where it really, really counts. And most importantly, that acetyl-CoA is what is going to go into the next step, the third step of cellular respiration, which is known as the Krebs cycle or the citric acid cycle. So it's important that you know that it's the acetyl-CoA that enters the citric acid cycle, again, also known as the Krebs cycle. Okay, so let's do a quick little run through up until this point. So up here, this is a schematic, okay? These are all 4 reactions that are going to take place in cellular respiration. Here we have glycolysis, which you know takes place outside of the mitochondrion. It is going to make a little bit of ATP and a couple of electron carriers. Then its end result is pyruvate, 2 molecules of pyruvate. Those 2 molecules of pyruvate are going to enter the mitochondria and are going to go through a reaction known as pyruvate oxidation, where they are going to make some carbon dioxide, a couple of molecules of electron carrier, and 2 molecules of something called acetyl coenzyme A. Note, pyruvate oxidation doesn't make any ATP, okay? Now, the next step is to take that acetyl-CoA and get it into the citric acid cycle or Krebs cycle, where again we're going to make, you see the pattern here hopefully, a little bit of ATP, maybe some electron carrier, maybe some more carbon dioxide. And when you look down here, you can kind of see those things taking place. Okay, so we're about to step through citric acid cycle in a little bit more depth. So the third reaction in cellular respiration is known as the citric acid cycle or the Krebs cycle. In the citric acid cycle, we are starting off with the end result of pyruvate oxidation, and that was acetyl-CoA. Now remember, acetyl-CoA is now a 2-carbon molecule. Remember, we got rid of one of those carbons when we did the pyruvate oxidation. So through the Krebs cycle, we're going to completely break acetyl-CoA down. We're going to break off every single carbon it has left and turn it into carbon dioxide. So at the end of the citric acid cycle, there is no carbon molecule left. The entire 6-carbon glucose has been broken up at this point, okay? So let's highlight that. All right, so the carbon dioxide that we are breathing out, that waste material, is literally formed through this process of oxidizing glucose, okay? There is a name for that process. It's known as oxidative decarboxylation, and it just means that we're removing that carboxyl functional group, that COOH functional group, okay? Now, in the citric acid cycle, we are going to make a lot more electron carriers. We're going to make both types, actually. Whereas in glycolysis and in pyruvate oxidation, we were only making NADH. We'll also make some ATP here, and it's going to be substrate level phosphorylation. So direct ATP making. And just like with when pyruvate was made and we had 2 molecules, because there are 2 molecules of acetyl-CoA, everything happens times 2. So The citric acid cycle is going to make 2 molecules of carbon dioxide, one for each carbon we have left. It's going to make 1 molecule of ATP through direct substrate level phosphorylation. It's going to make 3 molecules of NADH, and it's going to make 1 molecule of FADH2. Don't forget, both of these are electron carriers. So they are going to go to the electron transfer chain and help us to make more ATP then. So because everything is happening times 2, we're really in the citric acid cycle. For each glucose, we are making 4 carbon dioxides, 2 ATPs, 6 NADHs, and 2 FADH2s. This is the most stuff we've made in any of the reactions up to this point. So just to show you what I saved you from, this is what the citric acid cycle looks like in depth. There are a series of molecules made, but we call them intermediates because we don't make those molecules for long. We're just converting things and moving things and adding things, and then they become something else. But this is the citric acid cycle in all of its glory. Luckily, you don't need to know anything beyond what I've already taught you. Whoops. All right, ladies and gentlemen, so if you are keeping track, we're not doing an amazing job of making this ATP. Cellular respiration is all about making ATP. Where is it? Because we've made a couple of molecules in glycolysis, none in oxidative phosphorylation, and a couple of molecules again in the Krebs cycle. So what, we've made 4? There's no way a living system, even a simple living system, is operating on 4 ATP. You're right, there isn't. So there has to be some other like grand finale, right? Yes. And that grand finale comes in the form of the electron transport system, also known as the electron transport chain. Some people call it the electron transfer chain. It has a bunch of different names, but the same thing is true. It is the movement of electrons. And the movement of these electrons is going to allow us to do something we haven't been able to do up to this point. Now, every time we've made ATP prior to now, we've done so through substrate level phosphorylation, through directly taking a phosphate group off of a substrate and adding it on to ADP. In the electron transport chain, we make ATP through oxidative phosphorylation. So how are we going to do that? Well, it involves the use of high energy electrons. Well, where did those electrons come from? Well, we've been creating them every single time we created an electron carrier. Those electron carriers are carrying electrons in the form of hydrogen. So we're going to gather all of those up from the first 3 stages: glycolysis, glycolysis, pyruvate oxidation, and the Krebs cycle. And we're going to send them all to the folded inner membrane of the mitochondria. When it gets there, these electrons are going to be taken away from their carriers. The carriers are sent back so that they can pick up even more electrons. I always think of it as a bus or a shuttle system. And those electrons, once they get off the carriers, are going to be passed through a bunch of proteins that make up a transfer system. So as they pass, we're going to use their movement to pump, to establish, to create a hydrogen gradient. Okay, and a hydrogen gradient is just an area where there's a lot of hydrogen compared to an area where there is almost no hydrogen. So I'll draw you a quick diagram. So in my mitochondria is a double membrane organelle. The outer membrane is nice and smooth. The inner membrane is all folded up. Okay? And see that space that I have between my straight line and my wiggly line? That isn't by accident. That space is known as the intermembrane space. So these transfer systems that I'm talking about actually sit on the membrane. These proteins cover this entire membrane. So when the electrons start moving, I'm going to draw electrons as dots. Let's make them red. So when the electrons start moving from protein to protein to protein, that action gives us energy to move hydrogens into this space. So where are the hydrogens found? They're just found inside of the mitochondria. Okay, now this is happening along every single little tiny area of this folded membrane. Okay, so I'm just moving all these hydrogens in this area. And what's going to happen is I make this area where I have lots of hydrogens, and then in here I have very few hydrogens. So that's a gradient. We've already talked about gradients when we talked about transport over the cell membrane, right? So it's the same concept. Area of high concentration versus an area of low concentration. Now, there is one final protein who's very important. I'm going to draw him as green. And that protein is known as ATP synthase. All enzymes, for the most part, end with the ending ASC, so that tells you that this is an enzyme of some sort, but it's an enzyme complex because it's giant. Okay, so ATP synthase is going to allow these hydrogens to move back into the matrix. Think of what you know about passive transport. Things like to move from high to low concentration, right? So if I have a lot of hydrogen up here, eventually they're going to start flowing back down into the inner part of the mitochondria. And that happens across or through ATP synthase. So all these hydrogens will start going back this way, okay? As they are moving, indirectly, the energy that they create is going to be used to create ATP from phosphate and ADP. So an enzyme isn't specifically taking an ADP molecule and pushing a phosphate onto it. We have extra phosphate in the mitochondria. We have ADP in the mitochondria. The energy to cause them to bond is going to come from these hydrogens that are just moving through this giant enzyme complex. Okay, so this is what oxidative phosphorylation is, and this is why we describe it as indirect. Okay. All right, so let's see what we need to highlight. So over here, we're talking about our electrons came from our electron carriers. They pass through these carrier proteins. And then at the end, we make water. Oxygen is going to be the final acceptor of those electrons. And when the hydrogens combine with the oxygen, they make water. So this is why water is a product. Sorry about that. So this is why water is a product of cellular respiration. This is where the water is made. And as those electrons move, they lose energy as they go from protein to protein to protein. So eventually it's easy for them to just flow back into the matrix through that ATP synthase. And once they do that and they're put back with their hydrogen. I can't think of the word, y'all. I'm sorry. It's very early in the morning when I'm finishing this video. It's 5:44 a.m. Anyway, so when those hydrogens reconnect with their electrons, oxygen, because it's so electronegative, is going to be that final electron acceptor and create water. All right, this slide just tells you that even though carbohydrates are the main way that we do the typical steps of cellular respiration, Other alternative pathways also exist, and they will enter at different points in that those original 4 steps of cellular respiration. So you can use proteins to generate energy, you can use fats to generate energy. But your body's preference, of course, are your carbs. And to be honest, it tends not to process your fats very easily or quickly because your fats are considered storage. So carbs, proteins, and then fats in kind of that order. And then these are just the different places that those molecules will enter the original 4 steps. Okay, so how many ATP did we make? We've done all of this, but I still don't have a number, right? So let's go through and do the math. So about 32 ATP are indirectly made through oxidative phosphorylation using the electrons that you've been gathering, if you will, in the NADH and the FADH2 molecules. So on average, you make between 2.5 to 3 ATP for every NADH that's created in any of the 4 steps of cellular respiration. And you make anywhere between 1.5 to 2 molecules for every FADH2 that's created. So if we take into account how much direct ATP we make plus how much indirect ATP we make, this is the breakdown. So glycolysis makes 2 ATP directly through substrate level phosphorylation, and then it makes 2 NADHs, which would be the equivalent of about 6 ATP. Pyruvate oxidation makes 2 NADHs, because there's no ATP made directly, so again, another 6 ATP. And your Krebs cycle or citric acid cycle makes 2 ATP directly plus 6 NADHs, which is about 18 ATP, plus 2 FADH2s, which is about 4 ATP. Okay, so when you add it all up, grand total, if we kind of round up a little bit, we make about 38 ATP just through the 4 steps one time. I know that still doesn't sound like a lot, but remember that inner membrane is folded, and that's how life creates space. Okay, the mitochondria is still tiny, but it has this giant job that it needs to do. So it increases its surface area by folding. There are proteins literally covering every single section of that folded membrane. This 38 ATP represents what just one ETC can do. The mitochondria has thousands of ETCs all inside that folded membrane. So when you do the math, each mitochondria is not producing 38 ATP. It's producing 38 ATP times however many of these ETCs it has. And your body, each cell doesn't just have one mitochondria. It has as many as it needs. It just depends on what the energy requirement of that particular cell in that particular body part is. Now remember, once we get past glycolysis, everything else, pyruvate oxidation, citric acid cycle, electron transport chain, are all happening in the presence of oxygen. That's why we call it aerobic respiration. So what happens when there isn't oxygen? Well, we do an opposite process, something called anaerobic respiration, because ana means without. Okay, and in this case, I'm talking about the process of fermentation. So fermentation keeps ATP production going when oxygen itself is unavailable. Oops. What is wrong? Okay, there we go. So how exactly does this happen? Well, remember, glycolysis is technically anaerobic respiration. So glycolysis will still take place. Now, if oxygen is available, we're going to go ahead and do pyruvate oxidation, Krebs cycle, electron transport chain, all of the things. But if oxygen is not present, we're going to do fermentation instead. So NADH is going to bring electrons from glycolysis to an acceptor molecule. And then NADH is going to get converted back into NAD, which means that it can go back to the Krebs cycle and pick up more electron carriers. So think of it almost as we're banking electrons a little bit until oxygen becomes available. Okay? Now, there are two kinds of fermentation. There's alcohol fermentation, which yeast cells do, and the result of that is ethyl alcohol and carbon dioxide. And then there's lactate fermentation, which our bodies do, and this is carried out by fungi and bacteria as well. And in lactic acid fermentation, hydrogen atoms are added to pyruvate, and they form a new molecule known as lactic acid or lactate. Okay? And sometimes this is what's causing your muscles to feel super sore when you've worked out really hard or you've been running for long distances, that kind of thing. And your body will do this when you're working it very hard. So think running a race, or in my case, running from a dog, because Miss Courtney doesn't run. If you're a jogger, more kudos to you, credit to you. I don't understand that. We're just running to run. No, thank you. Anyway, but if you are, out for a run or whatever, you can only breathe in so fast. You can only get oxygen to your cells so quickly. So the longer you run, at some point your body has to compensate, because not every cell is going to get oxygen, which means some of your cells are doing fermentation and producing lactic acid. All right, this is where I leave you for cellular respiration, and we will pick up with photosynthesis next. Bye, guys.